Mitosis is one of the phases of the cell cycle. It follows G1, S, and G2 of interphase. During mitosis, the cell divides to create two genetically identical diploid daughter cells. The phases of mitosis include prophase, metaphase, anaphase, and telophase.
During interphase, DNA and organelles duplicate. At the end of interphase, the centrosomes divide and the mitotic spindle forms.
During prophase, chromatin condenses into chromosomes. The centrioles migrate to opposite poles, and microtubules form. The nuclear membrane also begins to break down.
During metaphase, kinetochore microtubules connect to the kinetochores of each chromosome. The chromosomes line up along the metaphase plate. In this phase, chromosomes are at their highest degree of condensation.
During anaphase, the non-kinetochore microtubules elongate. The kinetochore microtubules shorten, pulling the chromosomes apart and pulling the sister chromatids to opposite poles.
During telophase, cytokinesis occurs and a cleavage furrow begins to pinch the cell in two. Two daughter nuclei form, each with their own nuclear envelope and nucleoli. Chromosomes begin to decondense. There are now two genetically identical diploid cells.
Meiosis occurs in gamete cells. During meiosis, the cell divides to create four genetically unique haploid daughter cells. Meiosis is split into two phases, Meiosis I and Meiosis II.
During meiosis I, homologous chromosomes separate, and two genetically different haploid daughter cells are formed.
Before prophase I, the cell is in interphase and is considered a diploid cell. During the end of interphase, the DNA duplicates and the cell prepares to divide.
During prophase I, the chromosomes condense and the nuclear membrane breaks down. Homologous pairs line up during synapsis, forming tetrads, and crossing over occurs at the chiasmata, exchanging DNA. Centrosomes begin to move to opposite poles and the spindle forms.
During metaphase I, the homologous chromosomes line up along the metaphase plate as tetrads. Kinetochore microtubules connect to the kinetochore, which hold the two chromatids of the chromosomes together.
During anaphase I, the proteins responsible for holding tetrads together break down. This allows homologous pairs to be separated and moved to opposite poles by microtubules. Sister chromatids remain unseparated.
During telophase I, cytokinesis occurs and a cleavage furrow is seen. Each cell now has a haploid set of chromosomes. Each chromosome is still held together by the kinetochore. The nuclear membrane may partially reform and chromosomes may decondense.
During meiosis II, sister chromatids are separated. This process is essentially the same as mitosis and results in four genetically unique haploid daughter cells.
During prophase II, the spindle reforms. The nuclear membrane breaks down and the chromosomes condense and move towards the metaphase plate. They are still two chromatids connected by the centromere.
During metaphase II, the chromosomes line up on the metaphase plate. Kinetochore microtubules attach to the kinetochores of each chromosome, preparing to separate the sister chromatids. Note that the sister chromatids are not identical due to the crossing over that occurred in meiosis I.
During anaphase II, the proteins connecting the sister chromatids together at the centromere break down, and the sister chromatids are pulled apart to opposite poles by the microtubules. Once they are pulled apart, the sister chromatids are considered chromosomes.
During telophase II, cytokinesis occurs and a cleavage furrow is seen pinching the cell in two. The nuclear envelope reforms and the chromosomes decondense. At this point, there are now four genetically unique haploid daughter cells, and each chromosome contains only one chromatid.
To summarize, both mitosis and meiosis begin with diploid parent cells. However, mitosis results in two genetically identical diploid daughter cells, and meiosis results in four genetically unique haploid daughter cells.
Mitosis is used for growth, repair, and asexual reproduction in cells. Meiosis is used to produce gamete cells, reduce the number of chromosome sets by half, and create genetic diversity among gametes.